Wow.
1992.
Okay, wow. That was a while ago.
It was a ship on its way from Hong Kong to the United States. And they got released in the North Pacific.
Hmm.
So they would be really weathered.
Yes, they'd be in rough shape if they were out and about. A good book about the rubber ducks and how they've been used to study currents, absolutely, they're a great study of currents on accident, is called Moby Duck.
Oh.
So you can actually read about the whole situation in a book. That sounds really interesting. I never have enough time to read all of the books I would like to read.
There's another book called Flotsometrics and the Floating World, How One Man's Obsession with Runaway Sneakers and Rubber Ducks Revolutionized Ocean Science.
Ooh,
Sounds like a similar one.
that does sound like a similar one. Flotsam and jetsam is also fantastic. Are also fantastic words.
Yes. See more of those undercuts in the clay here, the little arches that form. It's a neat view in Argus. You can see a few of the steps in the clay. We're looking at one close up right now. Oh, and a birthday sponge.
That's a good looking sponge.
It is a fine looking sponge.
Zoomy zoom.
Oh,
Zoom zoom.
if you insist. So this looks like a saccocalyx, which is a glass sponge. So glass sponges take silicon and oxygen from the water and deposit it as, well, glass into glass rods that make a framework or the skeleton for the sponge. And the other cool thing about glass sponge, okay, there's a lot. Another cool thing about glass sponges is that they don't have cells. All of their cells are fused into one common cell membrane called a syncytium. So it's pretty cool. For a long time, it was wondered if glass sponges were always syncytial or if they were cellular and then became syncytial later, because other sponges are cellular. Thanks for the zoom. And it turns out that Sally Lees and Nicole Bourrienol in France together studied the development of Opsakis minuta, which is a small glass sponge, and they found that it was cellular up to the 32 or 64 cell stage of division, and then those cells divided unevenly into micromeres and macromeres that then fused together to make this syncytium.
With that zoom that we just had, is there any way for you to positively identify it without getting a sample?
Unfortunately, no. The spicules are sizes down to about 20 micrometers.
Okay.
So we won't be able to see it with the zoom, but more detail is nice.
Sure.
Yeah.
Okay.
All right, come up 5.
So stop me if I'm talking about sponges for too long, but here's another neat thing about the glass sponge.
No, let's learn.
Because
Nope.
it's syncytial, this is no stop.
We'll start doing dad jokes, Wayne.
keep talking.
Keep talking. So the syncytium means that all of the nuclei are housed within one cell membrane. So there's a lot of connectivity within the glass sponge. And so for other sponges, they don't have nervous systems, and so the only way that you can get cell-to-cell communication or coordination is by moving ions or other molecules. And so it's relatively a slow form of coordination that can happen between cells, compared to what a nerve cell can do, where it creates an action potential, so kind of a voltage that can shoot across a nerve. So sponges don't have nerves, they can't do that, but the glass sponge, because it's all connected with one single cell membrane, it can create an action potential, and that can move across that common cell membrane. And the end result is that the glass sponge can respond very quickly with a whole body response to changes in the water. So we did experiments where we kicked up sediment and looked at what the sponge did, and the sponge stopped pumping fairly quickly, and it's thought that it was able to stop its pumping by basically sensing and then propagating that action potential to stop all of the cilia or the flagella from beating.
Oh, that's pretty neat.
Super cool. It's like one giant. Okay, it's not like one giant nerve, but functionally that action potential is.
It is, and it's
Yeah.
kind of impressive to be able to keep up an action potential like that through I would say a semi-large organism, because that usually takes quite a bit of energy to do.
Yeah, and so having the syncytium is what makes that possible. So in us, the action potential is propagated by nerve cells, and they have these really long dendrites that project out, and so the signal can travel along those pathways. But going from one cell to another cell, even for nerve cells, you need to have special, they're called postsynaptic density molecules, to help facilitate that transfer. And so by being syncytial, the glass sponge gets around all of that and is able to propagate the action potential just with, well, I guess with ions, pumping ions across its body wall.
We have a question. Do glass sponges retain their shape if they are brought to the surface? I imagine that as glass they would. So they're pretty sort of a stiff. I would think they'd be very fragile because they are made out of glass, but
It depends on the glass sponge. So I'm not sure what saccocalyx feels like. It looks sort of crunchy, but it depends on how those skeletal elements are arranged. So in general, they're kind of like fiberglass. So you can bend fiberglass. It kind of bends like a sheet, but it has these glass fibers within it.
oh, okay.
So a lot of glass sponges are floppy, but fused or kind of stuck together like fiberglass. But then there's one that we've been studying at Sur Ridge that I've dubbed the marshmallow sponge because it's super, super soft, and when it comes up, it's just kind of a blob. And then there are some whose skeletons fuse secondarily, and those are very, very crunchy and would retain their shape. So there's a range.
Oh.
Ask me more sponge questions. I love this.
We will totally get to more sponge questions. I did mention the words flotsam and jetsam, and since someone had a question between flotsam and jetsam and what's the distinct meaning, usually we sort of say it as like there are odds and ends in the ocean. But it's interesting because under maritime law there's a distinction. Let me make sure I say this correctly. Flotsam is debris in the water that was not deliberately thrown overboard. So it may be a result of a shipwreck or an accident, or however it is, it wasn't deliberately thrown overboard. Jetsam is debris that was deliberately thrown overboard by the crew of a ship in distress, most often to lighten the ship's load. So that seems backwards.
There's also a third term that goes with that, but I can't remember what
There
it
is a third term. Where did it go? Laggin and derelict. And if my webpage would load, I would tell you what all of these things are. They're each different kinds of things that fall off a shipwreck or are part of a shipwreck. So it sort of depends on how these things were put or lost into the water as to who can reclaim them. So whoever discovers flotsam can kind of claim it. Jetsam, that was put overboard for sort of safety reasons, is technically still the owners. Legan refers to goods that are cast overboard and heavy enough to sink to the ocean floor, but are otherwise tied to a floating marker. And then derelict is goods that have sunk into the ocean floor and therefore abandoned, and there's not a lot of hope of reclaiming them. So there are lots of different types of things that come off or results of sort of shipwrecks. This brings me to NOAA's marine debris program. They actually have an app, and whenever you come into contact with any of these things, you can log what you're seeing, and it kind of helps us keep track of the different types of marine debris that are out there. Miles, would you like to do another explanation of what we're seeing with this sonar? It's still up in our quad cam, and we have some interested viewers.
Sure. Is there a starting point, or you just want me to?
Nope, just
ramble?
go for it.
Okay.
What is it? What is it doing? How is it helping us?
So the two main circles with the little spinny second hand, for lack of a better term, are the two Mesotech 360 scanning sonars, one on each vehicle. The left smaller circle is Argus, set at 20 meter divisions, so each red ring is 20 meters away from the vehicle, or at least away from the sonar head. On the right is Hercules at 5 meter division, so each ring is 5 meters away from the sonar head. And as it scans around, it's sending out an acoustic pulse and then measuring the return of that pulse. So it's sort of a range and bearing from the scanning sonar head, and the different colors denote sort of the intensity of that sound return. So you can see most of the circle is black, so it makes sense since we're up near this cliff. Away from the cliff is no return, and the cliff wall is showing up as a nice sort of bright yellow, high intensity return. And then the blue it drops off. We're sort of right at the top of this cliff, so we're sort of seeing the top of it, but it's also, as we descend, going to disappear on each pass.
Awesome. That makes sense to me. Viewers, let us know if you have any more questions. Thank you guys so much for tuning into Nautilus Live. We love having you join us. Your questions have been fantastic so far. Definitely sparked some thoughtful debate, and certainly we have learned a lot. If you are just joining us, we are currently cruising around the apron of Davidson Seamount. We are in Monterey Bay National Marine Sanctuary. This is an area that has yet to be explored, so we've never seen it, and we are currently exploring it. We're unsure of what we can find, but you guys are joining us in the adventure of seeing what's down here in the deep sea. Do we have a reading on what temperature or what the oxygen is down here at the moment?
Yes, we do. We're pulling it up.
Okay.
Alrighty, so what was your question? What did you want to know? Okay.
What was my question?
Oxygen.
Oxygen, thank you.
Oh, the current oxygen
Which
concentration.
is their oxygen and their temperature down there.
It's about 117.9 micromolars per liter.
Okay.
And temperature is 1.7 degrees Celsius.
Ooh, chilly.
To no one's surprise.
Yeah, that is a pretty common temperature that we've been seeing down here.
It's actually pretty common throughout the world.
Generally?
Yeah,
Oh.
yeah. The surface of the deep ocean bottom, 1.7.
So these are very neat clay steps that we're seeing here.
Is that a jelly? The Paralia?
Like a jelly.
There's a Paralia.
I want to poke it.
They just look done for.
Yeah, I think when they get
on
to
the
the bottom like that, they're living their last
Yeah.
Yahoo.
This looks like something snapped off.
Yeah. That's cool. Yeah, guys, I'm a very tactile learner, so it helps me to kind of touch things. And so exploring the deep sea is, I want to poke it. I want to touch it. Is it soft? Is it hard? Is it squishy? Is it slimy? I love learning all of those things.
You should definitely use all of your senses. I've been learning that with sponges, it's really helpful to smell them after we bring them up, too. Some of them have really characteristic smells.
Oh, that's interesting. Yeah, I think that's an important. We smell so many other things. Why not have identifying smells for creatures? I mean, you know, when you walk to a farm, like, it smells like farm, like
Yeah.
horses and
And you can
cows.
recognize some flowers by their smell. Like if you had your eyes closed and I put a rose in front of you,
Wow.
probably recognize that smell as the smell of a rose.
Yeah.
Is this,
Oh.
are these small rocks coral or shell?
I think a lot of these bumps, is that what you mean?
Yeah, just the tiny debris.
I think that's clay falling down.
Clay. Okay.
So, yeah, it's like some of these holes might be some of the clay that has fallen off this sheer wall. This is a tall one.
To the 5th graders from Virginia. Thank you guys for joining us. It took Hercules about 3 hours or so to get from the surface of the ocean all the way down to the bottom down here, and we are at a little over 3,000 meters.
It just keeps going.
It does keep going. It's a pretty tall feature. Oh yeah, and if we look at the isobaths, this is a very steep spot. Well, I mean, we could see that visually too. We can see it on the map.
The closer together the isobaths. So maybe we're plateauing out a little bit here.
A coral question. Have you found an increase in mercury in the corals? So we know that mercury can build up in some of our fish. Is that a thing with coral?
We haven't been measuring the mercury. We haven't been measuring the mercury in these corals. So we don't know the answer to
We lost
that.
our scientists for a sec, guys. They'll
But
come
we
back,
are-
don't worry.
we are
They're having
measuring-
a mini powwow.
The water while we're out here for persistent organic pollutants, such as PCBs. And when we get back to the lab, that water will be processed for a whole 200 plus persistent organic pollutants, otherwise known as POPs. So we hope to learn more from that. But that'll be in the water, not necessarily the corals.
Huh. Hello, Dave from Ohio. How long does it take Hercules to get back up from the bottom? About the same time. It's a little over 3 hours. We kind of sink down, and then our Argus is pulled up by the winch, and Herc kind of follows.
Yeah, ascent and descent rate is typically 15 to 20 meters per minute.
So there you go. We're giving you guys lots of math problems here this morning. Are our pilots on SPL?
Yes, we are.
How are you guys doing? Any fun facts to share? Seems like it's running pretty smoothly.
We're yo-yoing up and down over the terrain here, trying to keep up with the vessel.
So is it more challenging to drive because of that, or fly, I
It's a
suppose?
lot more fun.
A lot more fun. Well, that sounds like a good thing. Will you guys tell us a little bit more about sort of the challenges of flying? You said you're yo-yoing up and down. Is that due to the bottom here, our terrain that we're trying to look at? Is it the ship movement? Is it Argus? Is it the tethers?
Yep.
All of the above.
It's, we do a straight line with the ship, but the bathymetry is kind of zigging and zagging it in and out. So in order to maintain a visual on the seabed, which is the whole point of the dive, we're kind of going up and down with the terrain. But we don't know exactly what's here because this is a new town. So if we drop down too far into a box canyon, we can't get out fast enough with Argus. And we could rub up against one of these cliffs. So we're kind of using all the available information here, our sonars, the bathymetry, and just kind of the trends to stay out of trouble and keep a visual on the seabed. Yeah, sorry.
Gotcha, so it is a little bit of everything. You guys are very busy over there. Oh, thanks, Alex. That's a good comment. I will see if we can make that happen. They're suggesting that we have sort of someone with a lapel microphone during the recovery. So usually we don't have a lot of talking because there is a lot of coordinating between the ship and the crane and the ROVs, and so we don't want to get in the way. But if we were to be on a different channel or something like that, we might be able to get someone narrating what's happening. So we will consider that for next year. The question from Australia: what is the strangest human-made debris you have discovered at the bottom?
The strangest, well, I'll tell you some things that we've seen at Davidson, and you can tell me if they're strange. We have seen paint cans, we've seen buckets, we've seen brooms. We found a Coke bottle, a glass Coke bottle from Korea. That was fun to find. We found a old glass milk bottle from the Marin County area that maybe was produced during around World War II era. We found a newspaper that was still intact.
Wow.
From the Pacific Northwest. I think it originated from the Seattle area because we could see an ad from a department store that is special to Seattle. We found fabric. What else?
We found that Minute Maid soda can.
Yeah, that was just the other day.
Yeah, that label you guys thought was from the 70s?
I believe that was orange
Or the
soda.
90s.
Yeah,
90s.
orange soda, that's right. 90s, you said?
We're going to lose it for a minute, guys. The ship took a step to the north, so I'm going to have to chase it.
All right.
Tugged.
I saw a, not here, but I saw a purple shag carpet toilet seat cover.
Oh, well, that's fancy.
That's quite a find.
And the winner is?
In this Point Sur area, we found, which is close to an old military base, we found an old, now I'm going to forget the word, but it's a fake little torpedo bomb used for training exercises. That's not the technical term, but...
Oh.
What else have we found? So those are some of the crazy things we found. Luckily, they're not all in one place, and they're not all found at once. They are sporadically found over time.
We've come
Yes.
across ghost nets too.
Closer to
fishing
shore, we'll
nets.
see those. And we call them ghost fishing nets because although they're not fishing from a ship anymore, they are on the bottom still catching organisms on the seafloor. So we call it ghost fishing gear. And we have, the sanctuary has made an effort to try and remove some of that gear that's larger and maybe catching more things to stop that process.
Awesome. Yeah, that's a big issue. Yeah, as our fishing gear is made to be incredibly effective, and when no one is taking care of it or checking on it, it just ends up collecting and hurting a large portion of the marine life.
And of course, fishermen don't want to lose their gear, and they do their best not to, because it is worth money for them, and they don't want to screw up the environment.
Yeah,
So.
no, they're certainly not out to do that, hopefully. Yeah, and that would be an expensive process if you were constantly losing your fishing gear. Someone would like to know if descent and ascent time could be increased by using dry ice.
I don't know how that would work.
Let's see what else I got here.
I'll take a stab at that.
Solid form is heavy and transferred to gas form for ascension. Yeah, no, the solid form's not all that heavy. And when you put it in water, it sublimates with the release of the carbon dioxide. So we would be adding a lot of carbon dioxide into the ocean, which we're already doing too much of that, so we're not going to do any more of that. And it would be hard to kind of keep at its incredibly cold temperature. So we are down here for quite a bit of time. Our last dive was about 30, 35 hours. So to try to keep dry ice in a state where it would be functional for return, it would be far too energy costly. Antonella, I don't know if you are listening.
I am.
All right. Your mom and dad say hello.
Hi,
Coming
Mom
down
and
on the winch
Dad.
a bit, thanks.
Roger.
We've got a viewer from the Philippines. Oh, we got viewers from all over. Look at our numbers jumping. Hello, explorers. It is great to have you guys watching. Let's see, we've got Switzerland, Spain, Puerto Rico, Sweden, Norway, Italy, Canada, of course the US. I think we've got a number of fans, and I've certainly got some parents watching too, so thank you guys. We really appreciate your support, and we're incredibly excited to be able to share this exploration expedition with you. Somebody else was asking how we got here. So some of our scientists doing research, so I'm going to let them speak to how they got aboard the ship. But I actually applied for a fellowship, so we have internships and fellowships up on our website, and there are lots of different opportunities in different fields to join the Nautilus. So we applied for it. And scientists, what's your plan for getting aboard?
This is Erica Byrne from the Monterey Bay National Marine Sanctuary, and we're fortunate enough to have formed a partnership, the Sanctuary Program, with Ocean Exploration Trust. So for the last few years, we've been able to plan ahead and get ship time on the vessel. And I got on because I work for the Sanctuary as a research specialist. This is my first time on the Nautilus, which is very exciting. And we hope to be out here again in 2020. That's our hope.
I bet we can do it. I say we just stay out here forever.
And Erica is, I think,
Is it just me or the
uniquely
comms
expert
dead?
in Davidson Seamount as well, because
Can you hear me,
she
Wayne?
has put together a field guide along with other people recognizing the different fauna that have been found at Davidson Seamount in the past surveys. So you're the expert here.
Well,
for identifying.
yes.
The pseudo-expert.
My name is Amanda Kahn. I'm a assistant professor at Moss Landing Marine Laboratories. But last year, I was a postdoc at the Monterey Bay Aquarium Research Institute, and I was working in the benthic ecology lab of Jim Barry. who's collaborated with various people from the Monterey Bay National Marine Sanctuary. And so
I got you 6.
he was contacted about the cruise, and they know that there are a lot of deep corals and sponges that are at the top of Davidson Seamount, and so I am a researcher who has so far specialized in deep sea sponge biology. And so because we knew there'd be lots of sponges, I got to come along to see the sponges that are here.
And I'm Taylor Ann. I'm the ocean science intern. I also applied for this internship. I wasn't sure exactly what I wanted to focus on in my higher education after my undergrad, so I figured I had a lot of questions about the deep sea and was really curious about it, and was referred to apply for this internship to find out more and experience it for myself. And this has been the best learning opportunity. I feel like I'm learning so much. When I return back to school, all of my classmates I know are going to be asking about it, and I'll just fill them in on everything I've learned. And it's an incredible experience that I hope to continue on being immersed in. It's amazing that Ocean Exploration Trust actually makes this available to everybody and the public to view. You can learn a lot just from watching hours of these dives and listening to the scientists sharing their expertise and all the different species they know. So it's really interesting, and I hope I can come back.
Yeah, absolutely. So there are multiple different ways. You can always join us like you are here online asking questions. And it's great. We love to share our exploration with everybody who is interested. Do we use machine learning for automatic species recognition? Do any of you guys do that? I think so far a lot of us have been Googling and or flipping through species guides to try to determine what it is we are looking for, or what it is we are looking at. I know that that can be a thing, but it does take a very long time to calibrate, and you usually have to sort of make sure that you double check a lot of it. So I don't think any of us are currently using any sort of
You're on the north side.
machine learning
Faced.
for species identification.
There's a lot of research that's being done right now to train those models and to try to overcome the challenges with machine learning to recognize objects. So right now, the greatest expertise and the greatest accuracy is done with trained individuals who go through videos and identify. Some of the challenges are that if you look at a fish, for example, looking at it head on versus at an angle, it looks very different. It's a different shape. But also if the fish is sitting in front of a sea anemone or in front of any other kind of anything, or if there's something that's in front of part of the fish, it can create error in that machine learning. So I think it's something that's being developed. It's just still in the works, and there are a lot of different groups that are looking at that. MBARI is one. There is machine learning that's applied to other oceanographic data. So NOAA, I don't know the acronym. They're looking at coral reefs in Hawaii and the cover of different corals and being able to demarcate them and trace them using machine learning algorithms. And then there are also some machine learning methods applied to imaging equipment. So the imaging flow cytobot is one. I'm just going to look that up. I just saw a seminar recently that was about looking at the types of fauna, phytoplankton and zooplankton cells that were seen with the imaging flow cytobot and being able to partition them into different sizes and types of plankton.
That's really cool. It's a challenging system, but I do find it really interesting, and I think when we get something like that, really, if we're comfortable with really something like that, it will be a fantastic tool. Hello, all of you guys joining in. Oh, any positions for engineers? Absolutely. If you are interested in joining us out here on the Nautilus, we have a number of different intern positions and engineers that are on board.
would be the ROV internship.
Mm-hmm.
Seems like all the ROV interns are mechanical or electrical engineers. Nope, not Antonella.
Nope. Antonella, tell us more about you.
Sorry, most, I should say.
Well, officially my degrees are in computer science, but I do have kind of practical experience with mechanical engineering. I'm a roboticist at heart, and that involves anything from mechanical design and manufacturing to electronics and computing hardware, and then the software. So I kind of am in a jack of all trades, master of none position. But yeah, officially I'm a computer scientist, although that's debatable.
Gotcha. And you started out in the intern position.
I did, yeah. I was an intern last season. And so I think it's officially my 4th cruise back, 2nd season back. So I'm really excited to be back and learning a lot.
Awesome. How did you find out about the internship, and what suggestions would you give to someone who is interested in that position?
I got lucky. I go to grad school with my friend Jess, who's another Argus pilot, going to be coming on shift in a few hours. And she told me about it. And I met some people from Nautilus at the NOAA forum a couple of years ago and was encouraged to apply. I don't know how I would have found out about it otherwise. I got kind of lucky in that regard, but I think just kind of, for me, just having a diverse set of experiences, I think helped. Sort of a variety of engineering experiences and backgrounds rather than just pure computer science helped. I think that's probably why like not being a pure computer scientist has helped me quite a bit, I think, coming out here. So I think just building up as much as experience as you can in a wide variety of areas and applications.
Very cool. I love learning about different fields than my own. I've kind of been in the sort of biology field for a while, and this trip has given me the opportunity to talk to a lot of different career paths, and we all still love the ocean and marine exploration. But I don't know that much about engineering, so it's been fantastic to be able to talk to Antonella and many of the other engineers aboard. Well, keeping in that sort of realm, which vehicle is harder to control? So which ROV is harder to drive?
For me, Argus is far harder to drive.
And for me, Herc, so.
So I guess it depends on which one you drive more often.
Yeah.
Well, it kind of takes 3 people to drive Argus.
It does.
It does.
So Antonella has Yaw and Z for Argus, and myself and the bridge are sort of controlling the X and Y. So it's a big team effort.
There you go. So it takes a team to control these things. When we do come up, we are hauling it or pulling it back in with the cable. Do we have any ballast tanks on the ROV? I don't believe we do. We do? Yes? No.
The
Okay.
ballast tanks, did you say?
There's syntactic foam that has buoyancy, but it doesn't change.
Right.
Which ROV are you talking about?
Both. Either one of them, actually. So the question is, do you use compressed air blown ballast tanks?
Negative.
I would say
no.
no to compressed air because we are going into such pressurized depths. That sounds dangerous. But do we have, what kind of ballast tanks do we have? We have that syntactic foam. So on top of Hercules, there's a foam filled with little glass beads, and that is able to sort of help it deal with the pressure as a strong structure. And then words, I've lost them.
Well, do you
Right now there's 27 lead ballast blocks on the vehicle, so the ballast is arranged according to a rather long spreadsheet before the dive, depending on what kind of equipment is on the vehicle, and it's set up to be about 10 meters a minute positive.
Do we have any dive plates on Herc right now?
Yes, we do. We have 2 drop weights.
Okay.
They're commonly known in the trade as Alvin drop weights. They're just big metal weights that we can drop if we need to ascend faster than 15 meters a minute. We drop one of those, gives us another 5 meters a minute or so.
Yeah, and those plates are dropped with the robotic arm.
Oh,
They were actually
Correct.
lifted off and jettisoned.
just
They would become
jetsam.
Jetson? Or no, yeah, since
Maybe
there's no
jetsam.
tether with a float, it would be Jetson.
It would be jetsam because we're actively letting go of it to lighten our load.
You're
So
jettisoning
yes.
in it. Jettisoning.
Okay, time for shift change.
When are we going back to see the octopus? We don't know. We don't know yet. I'm not really sure what our plan for next year is. We are still, you know, kind of close to our octopus gardens, so we never know if we run into a few more octopods today. Remember, there's lots of different ways of saying plural of octopus. Octopuses, octopods, octopi. It is technically a Greek word. So octopi is putting a Latin ending onto a Greek word, so some people disagree with that. Octopuses is putting an English ending onto a Greek word, so some people disagree with that. And then there is octopodes, which is kind of sticking to sort of the family Octopoda.
So you're saying octopus is plural and singular, like moose.
I feel like that's easier for everyone, but
But I think we tend to call them octopods and not octopuses.
yes.
So the Beatles were all wrong?
Beetles were wrong.
Oh no. But that's like a plural, right?
You
Because it's
just made
the
a bunch of enemies.
octopuses garden. Like multiple octopods? Or is it like a, this is my garden? I'm going to leave that one
Yeah, leave it.
for you guys to debate. So recently, there was a PBS documentary about a dreaming octopus. I have yet to see it. Have any of you guys had a chance to see that show yet?
Not the show, but I've seen the clip
You've seen
where
the
the
clip?
octopus turns various colors while it's sleeping.
Yes, while it's sleeping. What do you think of this recent discovery?
Well, I think there's more to learn. I don't know if it's conclusive. But it is fascinating if that's true that they dream.
Ship is stopped and waiting for him.
I wonder if other
Ship is
things
stopped
do the
and
same
waiting for
thing.
him.
Yes, I agree with that. I think it would be very interesting to continue that sort of exploration into deep sleep and dreaming and other creatures. And if it's dreaming in the sense that we're dreaming, or different sort of just synaptic nerves firing or something like that. We would like one more fact, or a few facts, about siphonophores.
You want a fact, or you're going to provide a fact?
There is a request for facts.
Oh, well, I wish
about
Amanda was here. She
siphonophores.
stepped out.
Oh no. Well, we'll get you those siphonophore facts when we can, you guys.
So what we're looking at now are some anemones and what might be a grenadier.
Lying on a vertical face of a cliff.
Yeah. It's mostly clay, we think.
Okay.
Did we previously give siphonophore facts that I need to be caught up to speed on? Ah, siphonophores are one of the largest
morning.
living organisms
I
in
think
terms
I'm
of
ready.
a colony in the ocean.
All right, guys, good luck with your facts. Keep up the fantastic questions. I got Megan here to keep you going. Thank you guys so much for joining us on Nautilus Live.
Hey everyone, watch change underway. Get some new folks on headsets. This is a great time to hit refresh on your browser. Dumbo Octopus. Dumbo Octopus.
Wow.
Hello.
Yeah. I was like, I don't know if you're on
Welcome to the
headsets. I'm going to start shouting.
Oh, how beautiful.
We're middle of a shift change, but
Yeah.
that looked fabulous.
What is the, do we know
I
what
thought
the
it was
genus on this?
Vampiroteuthis, but maybe it's, what is it?
It does look more like that.
Look at those.
Little
Wow, he's so
eyes.
beautiful.
Yeah,
I think
His
that's
eyes.
Vampiroteuthis.
Vampiroteuthis.
that's okay.
Could definitely tell how they got that Dumbo name.
How are you doing, Greg, as far
There's
as
two fins on the side.
pull goes?
Amazing.
Yeah.
Wow.
I like how
Doing
he
a
waited to
dance.
pounce on us until the moment we weren't ready. Watch change.
Wow.
That's beautiful.
That is so awesome, you guys.
Yeah. Yeah.
I think it planned it.
How incredible.
Cool.
You can see, I mean, look how efficient it is. It can
Yeah.
just sort of hang in that posture.
Neutrally buoyant.
Cirrothamna magna, I might have butchered that, but thank you so much, Mike, one of our go-to cephalopod experts, one of our scientists ashore.
So
Such a help to
Yeah,
us. Thank you.
we're in the process of looking this one up. We're not
Mike
100%
just
sure.
said Cirrothamna magna, so
Yeah, we're
he's
not
our
100%
go-to
sure. That's a
I'm
vampire
curious
toothus.
about those wings, if they're like a derivation of the fins on squid. Where did those come from? What embryologically, what's the source?
Getting pulled.
Definitely getting some good shots here. Great camera work.
And look at the dexterity in those wings. It's just magnificent.
Lots of surface area there, just hanging. So
All right,
cool.
video, let's come full wide. That's all we've got time for. We gotta go in
All
the
right,
other direction.
bye-bye.
Thank you.
Bye, have a nice day.
See you next time.
Thanks for visiting.
So you're settling, Argus is settling towards
I'm
the
awake
ship.
now.
Yeah, there you go.
That was so awesome. The prior shift is coming back because we want another octopus.
So that is not
Yeah,
strictly the
expect they'll be able to
Dumbo
reacquire it.
octopus, is it?
No.
Megan, what did you say the person wrote in and said?
Cirrothamna magna.
Cirrothamna magna.
Okay. That's
And
what's
serrate fins
yeah.
are homologous to squid fins.
and what?
You asked embryologically where they
Mhm,
develop from, and serrate fins are homologous to squid fins.
Okay.
From our same scientist
Okay.
ashore. Thank you
Great.
for helping
Yeah,
us.
that's what's so
These moments where we get
amazing.
excited and lose our
Yes,
minds. Thanks for
yes.
bringing us back with
Well,
the science facts.
And we put our questions out, and the community out there helps us answer them.
Yes.
Okay.
So we're just reacquiring the wall here. We let Argus swing a bit while we were looking at that great video footage there.
That's okay.
Yeah.
Should I change my heading or stay?
So we're gonna eventually go up around this way. But for right now, well, he's facing south right now. So once you get situated.
Oh yeah. Yeah, you're on down cam now.
Yeah.
Okay.
So a little more about that. Originally, this species was placed in the Cerateuthis group in 1932 when it was described, but in 1987, researchers and specialists, taxonomists questioned that, and by 1999, by looking at the internal shell and reordering the group, O'Shea in 99 moved it to the Cerathama
There we go, cool
genus.
shot there.
So it actually changed, which is
got more.
really cool.
Yeah.
1986 was the original, or was the second description of the species from the holotype, but it was kind of mashed up. I imagine in that day it was probably caught by a trawl or by fishing. So to name a new species and to describe them, you have to get a voucher specimen. Then that work is done by specialists who know the group of animals particularly well. So, one of us doesn't get to just like name a species or watch it zoom by the camera and say, I think that's probably new. It gets sent off to specialists to examine.
1932 this.
So is this at the top of this cliff?
Not quite sure if that's the top ledge.
It's interesting to see all the cnidarians just kind of spread along that upper
looks
band.
like there's some more.
Okay, well then that's even more amazing that they're right in that band.
It seems to be wherever there's a vertical or overhang, and not sediment.
Right.
They're happier, which makes sense. It's a little bit harder.
So here's a question. Do the larvae choose those locations, or do the...
Maybe we're just looking at survivorship bias.
Survivorship bias, or can they actually move? Some anemones do move.
If he's south, and I want to go that way. That's 135 relative, which is about 315. What's that look like over here? 3. Should we do it? Try some 315? I'm just going to do a couple of steps, I think. I don't know about tracking. In case we overshoot it. Paul, can you rotate your heading to your starboard?
Yeah.
We'll just follow Herc around as it tracks along, and I'll give a couple of steps here. Bridge nav. Can I have a step 50 meters bearing 315? Yes, thank you.
So Rennie, do you want to give us a little bit of the big picture perspective? Where are we along this feature versus where we were last night? How much do we have left to go?
Sure. Yeah, we're still on this this kind of apron lobe that's coming off of. It's to the southwest of Davidson Seamount. We're targeting it because it's a steeper slope. There's two kind of roughly two planes. And the interface between them has this steeper slope that we were curious if it was going to host
Aaron,
some
push in.
organisms, if it was a harder substrate. It looks like it's been mostly this hard sedimentary.
Ooh.
That's a plastic bag.
That is a plastic bag, with trash inside.
Yeah.
Wow. Got a 7-Up can.
Including a 7-Up can.
Yeah.
It's a new habitat now.
Wow.
Ugh.
And we're generally heading towards a cone-like feature. that rises at least 200 meters off the seafloor.
Jamie, I know you're already on it, but I love getting captures of these. I use them in a lot of my presentations.
Yep.
Yeah, there's some Bersingids on there.
I think this would be a powerful one.
Mm-hmm.
Wow.
Now it's, yeah, so we're just going, back row, we're just going to go in this kind of circ here, this feature. It might be a little bit slow just because it's not a long straight pass, so we'll come around here, and then it's up to you if we want to keep following this contour around in here and then head to this section and then up to the cone. It looks like that was the original dive plan, so just let me know if you want to divert from that, otherwise I'll continue on that.
Well, yeah, for now we'll continue from point 12 to 13, and then Chad King will be back in the room in a bit.
Sure. Sounds good.
We're ready. Oh, ship is moving.
Ship is moving, yep.
That main comp was the only weird thing.
Let me see the numbers for the...
So you're facing 225. Yeah, so 315 should be all right for now. You're sinking a bit, just in case.
I think it was 3 about 24 hours ago during our first shift. That was even... Did we even launch?
I was in the crane for launch.
Right, never mind.
That's a good excuse. I was in the crane. I didn't see it. So how deep is this drop off right here? How much water do we have beneath us?
Well, right now, it's an excellent question.
I don't know if you can tell if you're kind of edged up on it.
The map says the entire feature.
Argus is 50 meters off bottom and 25 meters above Herc.
Okay.
Yeah, I was going to say the whole feature on